Block 1, Unit 4, Antibiotic Resistance. The successful use of any therapeutic agent is compromised by the potential development of tolerance or resistance to that compound from the time it is first employed. This is also true for agents used in the treatment of bacterial, fungal, parasitic, and viral infections, and for treatment of chronic diseases such as cancer and diabetes. It applies to ailments caused or suffered by any living organism, including animals, fish, plants, and insects. A wide variety of biochemical and physiological mechanisms may be responsible for resistance. In the specific case of antimicrobial agents, the complexity of the processes that contribute to emergence and dissemination of resistance cannot be overemphasized. And the lack of basic knowledge on these topics is one of the primary reasons that there has been so little achievement in the effective prevention and control of resistance development. Most international, national, and local agencies recognize this serious problem. Many resolutions and recommendations have been proposed, and numerous reports have been written, but to no avail. The development of antibiotic resistance is inevitable and relentless. The most striking examples, and probably the most costly in terms of morbidity and mortality, concern bacteria. The discovery of these infectious agents in the late 19th century stimulated the search for appropriate preventative and therapeutic regimens. However, successful treatment came only with the discovery and introduction of antibiotics half a century later. Antibiotics have revolutionized medicine in many respects, and countless lives have been saved. Their discovery was a turning point in human history. Regrettably, the use of these wonder drugs has been accompanied by the rapid appearance of resistant strains of bacteria. Antibiotic resistance refers to the ability of the bacteria to resist the effects of one or more antibiotics. So despite treatment, some bacteria survive and divide to create a new population of bacteria that are resistant to the first antibiotic or class of antibiotics used. Bacteria that are resistant can infect different people, leading to an infection that cannot be treated with an antibiotic that is thought to be effective. Bacteria may be inherently resistant to antibiotics. For example, an organism lacks a transport system for an antibiotic, or an organism lacks the target of the antibiotic molecule. Or, as in the case of gram-negative bacteria, the cell wall is covered with an outer membrane that establishes a permeability barrier against the antibiotic. Several mechanisms are also developed by bacteria in order to acquire resistance to antibiotics. This can occur through transfer of resistance genes from one bacterium to another. Each of these mechanisms requires either the modification of existing abnormal or mutated genetic material, DNA, or the acquisition of new mutated genetic material from another source. Medical pundits are now warning of a return to the pre-antibiotic era. A recent database lists the existence of more than 20,000 potential resistance genes, R genes, of nearly 400 different types, predicted from available bacterial genome sequences. Antibiotic discovery, modes of action, and mechanisms of resistance have been productive research topics in academia, and until recently in the pharmaceutical industry. As natural products, they provide challenging intellectual exercises and surprises with respect to their chemical nature, biosynthetic pathways, evolution, and biochemical modes of action. The total synthesis of such natural products in the laboratory is difficult and costly, since these small molecules are often extremely complex. Rapidly developing resistance, combined with an historic decrease in interest from pharmaceutical companies in developing new antibiotics, is leading to a critical problem in treating bacterial infections that were previously successfully cured with antibiotics. Unfortunately, the colossal need for these valuable drugs has had a significant environmental downside. In the 60 years since their introduction, millions of metric tons of antibiotics have been produced and employed for a variety of different purposes in patients, agriculture, and industry. Improvements in production have provided increasingly less expensive compounds that encourage non-prescription and off-label uses. The planet is saturated with these toxic agents, which has, of course, contributed significantly to the selection of resistant strains. The development of generations of antibiotic-resistant microbes and their distribution in microbial populations throughout the biosphere are the results of many years of unremitting selection pressure from human applications of antibiotics via underuse, overuse, and misuse. This is not a natural process, but a man-made situation superimposed on nature. There is perhaps no better example of their Darwinian notions of selection and survival. In other words, the natural rate of mutation in bacteria has inadvertently led to a high rate of resistance in nature because there may be a selective advantage to being resistant to antibiotics. As a reminder, the term antimicrobial refers to any compound that can either stop the growth of or kill microorganisms, including viruses, that exploit the biology of other organisms. The category includes agents that affect bacteria, viruses, parasites, and fungi, and these agents often work in more than one of these categories. Antibacterial soap, for example, can kill bacteria and funguses. Likewise, some drugs that are approved as antifungals are also effective antibiotics. Studies with these compounds have often shown unexpected antibiotic effects that indicate a variety of other biological activities. The result has been a significant number of additional therapeutic applications of antibiotics as antiviral, anti-tumor, or anti-cancer agents. In some cases, the alternative applications have surpassed those of antibiotic activity in importance, such as in the treatment of cardiovascular disease or use as immunosuppressive agents. The definition of antibiotic, as first proposed by Selman Waxman, the discoverer of streptomycin and a pioneer in screening of soils for the presence of biologicals, has been seriously overinterpreted. It is simply a description of a use, a laboratory effect, or an activity of a chemical compound. It does not define a class of compound or its natural functions, only its application. The generic term antibiotic is used here to denote any class of organic molecule that inhibits or kills microbes by specific interaction with bacterial targets, without any consideration of the source of the particular compound or class. Thus, purely synthetic therapeutics are considered antibiotics. After all, they interact with receptors and provoke specific cell responses and biochemical mechanisms of cross-resistance in pathogens. Since the introduction in 1937 of the first effective antimicrobials, namely the sulfonamides, the development of specific mechanisms of resistance has plagued their therapeutic use. Penicillin was discovered by Alexander Fleming in 1928, and in 1940, several years before the introduction of penicillin as a therapeutic, a bacterial penicillinase was identified by two members of the penicillin discovery team. The suffix ase refers to an enzyme that acts on a molecule or compound. Once the antibiotic was widely used, resistant strains capable of inactivating the drug became prevalent, and synthetic studies were undertaken to modify penicillin chemically to prevent cleavage by penicillinase, beta-lactamase. The enzymes break bonds in the chemical structure of penicillin that are also present in other molecules of the same class of antibiotics, the sulfonamides. The Dark Ages and Primordial Era describe pre-antibiotic development. During this time, some indigenous populations recognized the utility of antibacterial plants, but antibiotics were not formally developed until the 1930s. During the golden years, most of the antibiotics used today were discovered. Antibiotics discovered during this time were easily identified from compounds that occur in nature, and resistance was also widely recognized. After all easy-to-find antibiotics were made, more sophisticated approaches that required more effort and money were necessary. This led to the lean years, when antibiotic discovery decreased dramatically. We are currently in this phase, and even more challenges to address mechanisms of antibiotic resistance are occurring as resistance is observed very rapidly after a new antibiotic becomes available. As resistance occurs more quickly, pharmaceutical companies are less incentivized financially to invest large amounts of money in their development because the drugs are maximally effective for a short period of time. Multi-drug resistant MDR forms subsequent to antibiotic use. For example, MDR M. tuberculosis is a major pathogen found in both developing and industrialized nations. Other serious infections include nosocomial, hospital-linked infections. The term superbugs refers to microbes with enhanced morbidity and mortality due to multiple mutations endowing high levels of resistance to the antibiotic classes specifically recommended for their treatment. The therapeutic options for these microbes are reduced, and periods of hospital care are extended and more costly. In some cases, super-resistant strains have also acquired increased virulence and enhanced transmissibility. Tuberculosis is the archetypical human pathogen. It evolved with the human race and currently infects as much as 1/3 of the world population. While the groundbreaking discoveries of streptomycin and isoniazid provided vital treatments, resistance development was rapid. The introduction of cocktails of anti-TB drugs has become an essential treatment regimen with considerable success. However, for a variety of reasons, multi-drug resistance continues to compromise TB therapy throughout the world. M. tuberculosis strains resistant to 4 or more of the frontline treatments, for example, extremely drug-resistant XDR strains, have appeared and spread rapidly in the last decade or so, and now there are TDR, XXDR strains which are totally drug-resistant. Concerning hospital-acquired diseases, Pseudomonas aeruginosa has evolved from being a common burn wound infection to a major nosocomial threat. In this case, again, antibiotic resistance mechanisms evolved completely by chance with the introduction of new antibiotic derivatives, compromising the most effective treatments. P. aeruginosa is of considerable concern for patients with cystic fibrosis. The pathogen is highly persistent and can avoid human immune defenses. Currently, the most notorious superbug is the gram-positive organism Staphylococcus aureus. S. aureus has a close association with humankind. It is carried in the nasal passages of 30% of the population, and its presence has long been linked to common skin infections such as boils. It does not have the historical reputation of M. tuberculosis, but in recent years, this multi-drug resistant pathogen has emerged as a major nosocomial infection. The landmark discovery and introduction of methicillin, the first designer anti-resistance antibiotic, in 1959 were thought to be a sure defense against the penicillinases, but the appearance of a methicillin-resistant S. aureus, MRSA, or MRSA, within just 3 years led to other multi-antibiotic resistant variants, and the acronym now denotes multi-drug resistant S. aureus. Relatively recently, MRSA has moved outside the hospital and became a major community-acquired CA pathogen with enhanced virulence and transmission characteristics. A long-recognized hospital denizen, the toxin-producing anaerobe Clostridium difficile, or C. diff, is increasingly found as the cause of severe intestinal infections. Recently, hypervirulent toxin-producing strains have been recognized. Being a gram-positive spore former that can be spread through the air, it's a hardy organism and is readily transmitted by hospital personnel on equipment and as aerosols. Its renewed prominence is considered the result of extensive hospital use of antibiotics, such as expanded spectrum cephalosporins, the newer penicillins, and fluoroquinolones that cause significant depletion of the gram-negative intestinal microflora, thus enhancing C. diff colonization. In other words, these infections are the direct result of antibiotic use. With respect to the global control of endemic and pandemic infectious diseases, a significant problem is the availability of reliable systems for tracking outbreaks of serious infections. Despite the heroic efforts of the World Health Organization, such reporting is non-existent in many parts of the world. A lack of information concerning the early stages of an epidemic bacterial infection has retarded appropriate remedial action in many cases. Microbes such as bacteria, viruses, fungi, and parasites are living organisms that evolve naturally over time through random mutation of DNA that may be beneficial to their survival. Their primary function is to reproduce, thrive, and spread quickly and efficiently. Therefore, microbes adapt to their environments and change in ways that ensure their survival. If something stops their ability to grow, such as an antimicrobial, genetic changes can occur that enable the microbe to survive. There are several ways this happens. Natural biological causes include selective pressure. In the presence of an antimicrobial, microbes can either be killed or, if they carry resistance genes, survive. These survivors will replicate and their progeny will quickly become the dominant type throughout the microbial population. Mutation. Most microbes reproduce by dividing every few hours, allowing them to evolve rapidly and adapt quickly to new environmental conditions. During replication, mutations arise randomly, and some of these mutations may help an individual microbe survive exposure to an antimicrobial. Gene transfer. Microbes may also get genes from each other, including genes that make the microbe drug resistant. Bacteria multiply by the billions. Bacteria that have drug-resistant DNA may transfer a copy of these genes to other bacteria. The process of DNA transfer from one bacterium to another is random, so some DNA may not be beneficial to survival in the presence of the antibiotic, while other DNA may be helpful. Non-resistant bacteria receive the new DNA and become resistant to drugs. In the presence of drugs, only drug-resistant bacteria survive. The drug-resistant bacteria multiply and thrive. Societal pressures. The use of antimicrobials, even when used appropriately, creates a selective pressure for resistant organisms. However, there are additional societal pressures that act to accelerate the increase of antimicrobial resistance. Inappropriate use. Selection of resistant microorganisms is exacerbated by inappropriate use of antimicrobials. Sometimes healthcare providers will prescribe antimicrobials inappropriately, wishing to placate an insistent patient who has a viral infection or an as yet undiagnosed condition. Inadequate diagnostics. More often, healthcare professionals must use incomplete or imperfect information to diagnose an infection and thus prescribe an antimicrobial just in case, or prescribe a broad-spectrum antimicrobial when a specific antibiotic might be better. These situations contribute to selective pressure and accelerate antimicrobial resistance. Hospital use. Critically ill patients are more susceptible to infections and thus often require the aid of antimicrobials. However, the heavier use of antimicrobials in these patients can worsen the problem by selecting for antimicrobial resistant microorganisms. The extensive use of antimicrobials and close contact among sick patients creates fertile environment for the spread of antimicrobial resistant genes. Agricultural use. Scientists also believe that the practice of adding antibiotics to agricultural feed promotes drug resistance. More than half of the antibiotics produced in the United States are used for agricultural purposes. Antibiotic-resistant strains of bacteria that originate on farms and are accidentally present on foods sold in grocery stores causes multi-state outbreaks that have led to deaths, particularly of the young or elderly consumers. Most, but not all, resistance mechanisms are encoded by plasmid DNA, which is potentially transmissible to other bacteria. The most common mode is enzymatic inactivation of the antibiotic through altering the chemical structure of the antibiotic. A cellular enzyme may acquire a DNA mutation that enables it to react with the antibiotic in such a way that it no longer affects the microorganism. An alternative strategy utilized by many bacteria is the alteration of the antibiotic target site so the antibiotic can no longer work in the cell. Efox pumps are high-affinity reverse transport systems located in the membrane that transport the antibiotic out of almost all mammalian cells. Efox pumps naturally remove toxins and other substances that may harm the cell. DNA mutations may increase the efficiency of these pumps to selectively pump antibiotics out of the cell or increase the number of these pumps on the membrane to increase the rate of transport. The development of resistance is inevitable following the introduction of a new antibiotic. Initial rates of resistance to a new antibiotic are normally on the order of 1%. However, modern uses of antibiotics have caused a huge increase in the number of resistant bacteria. In fact, within 8 to 12 years after widespread use, strains resistant to multiple drugs become widespread. Multiple drug-resistant strains of some bacteria have reached the proportion that virtually no antibiotics are available for treatment. Antibiotic resistance in bacteria may be an inherent trait of the organism. For example, a particular type of cell wall structure that renders it naturally resistant, or it may be acquired by means of a mutation in its own DNA or acquisition of resistance-conferring DNA from another source. Vertical gene transfer. Although mutation is a very rare event, the fast growth rate of bacteria and the absolute number of cells attained means that it doesn't take long before resistance is developed in a population. Once the resistance genes have developed, they are transferred directly to all the bacteria's progeny during DNA replication. This is known as vertical gene transfer or vertical evolution. The process is strictly a matter of Darwinian evolution derived by principles of natural selection. A spontaneous mutation in the bacterial chromosome imparts resistance to a member of the bacterial population. In the selective environment of the antibiotic, the wild-type non-mutants are killed and the resistant strain is allowed to grow and flourish. Horizontal gene transfer. Another mechanism beyond spontaneous mutation is responsible for the acquisition of antibiotic resistance. Lateral or horizontal gene transfer, HGT, is a process whereby genetic material contained in small packets of DNA can be transferred between individual bacteria of the same species or even between species. There are at least 3 possible mechanisms of HGT, equivalent to the 3 processes of genetic exchange in bacteria. These are transduction, transformation, or conjugation. Transformation is the process where parts of DNA are taken up by the bacteria from the external environment. This DNA is normally present in the external environment due to the death and lysis of another bacterium. Transduction occurs when bacteria-specific viruses, bacteriophages, transfer DNA between 2 closely related bacteria. Conjugation occurs when there is a direct cell-cell contact between 2 bacteria, which need not be closely related, and transfer of small pieces of DNA called plasmids take place. This is thought to be the main mechanism of HGT. The learning goals for this unit, antibiotic resistance, are: list the main causes of and their relative contributions to antibiotic resistance in the United States. Describe 3 cellular mechanisms of antibiotic resistance. Explain the clinical significance of antibiotic-resistant bacteria. Draw mechanisms of bacterial transfer of antibiotic resistance-related genes.